Cell membranes are critical for life. They separate the compartment that we can maintain homeostasis separately from the disorder of our environment. And we are developing the first breakthrough new class of small molecule treatments that can repair the holes in cell and cellular membranes which are formed by misfolded proteins in many degenerative diseases. So actually at least 50 deadly degenerative diseases now associated with protein misfolding and aggregation. The biggest ones you see here. And actually our vision is to develop cures for 6 major diseases, starting with our primary focus on Parkinson's disease. But together these diseases affect over 500 million people at a cost of $3 trillion a year. And there are no effective disease-modifying treatments for any of them, and rapidly growing problem with the aging population. Now, in every single one of these diseases, a specific protein or peptide, or sometimes more than one, aggregates and misfolds into insoluble fibers, and for years the pharma industry has been trying to target those without any effect, bringing into question the whole idea as how is this related to disease, if at all. And it turns out that the intermediates of protein aggregation, soluble oligomers, are incredibly toxic to cells, and so companies are now trying to develop preventative treatments to stop their formation. We take a different approach. We ask, why are these so toxic to cells? And it turns out in every single case, these misfolded proteins, different diseases, different parts of the brain or body, they all form, embed into cell and cellular membranes and permeabilize the membranes and allow uncontrolled influx of calcium, loss of homeostasis, and that ultimately results in loss of control and degradation and cell death and degeneration. So we are the only company that is specifically targeting the actual problem, which are holes in the cell membrane, rather than the upstream cause of these diseases. So in doing so, by targeting the holes, imagine you got a hole in your tire, the solution is to fix the hole in your tire and pump it up, not to clean the glass and nails from the road. That's preventative, but it's not going to help you if you're already developing the disease. So by targeting the holes in the cell membranes, we can repair the cell membranes and allow the cells to fully recover and restore homeostasis. And therefore, our goal is not just to prevent the progression of disease, but to reverse it and cure it. So actually, amyloid pores are very similar to bacterial pore-forming toxins, as well as antimicrobial peptides. Bee and wasp venoms also form pores. Holes in cell membranes are always bad news, and you can actually see these things by electron microscopy, atomic force microscopy from disease-related tissues. So we know they're there. In fact, amyloid pores across all these diseases are now supported by hundreds of peer-reviewed papers over the last 30 years across all these diseases. And so the question is, why is no one else targeting this? Well, actually, it's a very challenging target because they're dynamic, metastable, heterogeneous, and to most medicinal chemists, it would be like shooting a moving target that you can't see. So why are we doing this? Well, we've been able to overcome this problem and identify a class of molecules that can successfully block and effectively block these pores. But there are other barriers which we've also been able to address, and actually these are mostly due to cognitive biases in human behavior. So this is what makes us stand out as having a unique technical solution as well as the mindset to be able to address risk and uncertainty properly. So how are we doing this? We've actually developed 3 completely new classes of molecules. The first 2 are in active development right now, which can block and dissolve the pore structures within the cell membranes. And then we have a second generation of conceptual treatments which will piggyback on the first 2 that will allow targeted protein degradation of these pores, specifically based on the structures. So just to give some science, I mean, we start with molecular docking. We have a unique molecular architecture that is uniquely able to block these pores. And we show that only our design of molecules can do this and fully block the pore cavity and bind much more strongly and tightly than anything in the, known in the chemical virtual database space. We also run very fundamental liposome-based assays. So that emulates the cell membrane. And what you find is that when you add the protein misfolded oligomers, you get the pore formation, and we can measure the leakage of dyes through these pores. And we can use that to screen and show that which inhibitors are actually able to block those pores. And we then run dose response curves on those. And so far, I mean, these show inhibitors working down to 7 nanomolar. We now have identified inhibitors working at 1 nanomolar. And actually, there's a very good correlation between the in vitro activity that we see in the pore blocking activity and the molecular docking. So we know we've got a good predictive model with very strong and clear structure-activity relationships to be able to further optimize the chemistry. We've also validated these in cell assays with a clear dose response showing protection at substoichiometric concentrations relative to the pore-forming peptides, and able to fully recover and reverse the toxicity of these pore-forming proteins, alpha-synuclein in this case. Moreover, our initial prototype Amporin, that's the Amporin, the pore dissolving agent, we found that with a single oral dose, was able to fully reverse the neurological deficits in 2 mouse models, Alzheimer's on the left, Parkinson's on the right, within hours of treatment. And that kind of effect has never been seen before. And we think that's because of the mechanism. By targeting the pores, repairing the membrane, we're able to restore the full physiological function of neurons which are not yet dead. Once they're dead, they're dead. Now, our plan going forward, as I say, we're targeting Parkinson's primarily, and then have a plan to kind of follow up expanding out into other diseases. And then obviously, depending on who's paying our bills in terms of funding, we can move those around. But Parkinson's is the core indication. We're focused on finalizing lead optimization and validation, looking for 2 to 3 million francs seed funding to get into IND enabling studies. But our business model, because we're looking for targeted disease reversal, acute disease reversal, we can get clinical proof of concept within weeks or months rather than years, where you try to measure delay in chronic progression. So we would aim to license out a proof of concept in phase 1B, or at the latest after phase 2A, by which time we'll also have an exit. Now, I won't go into this, but we have a screening platform that allows a kind of cascade of optimizing our compounds, supported by mechanistic supporting studies. And then there's a whole pathway that we can follow to develop a full data package for in vivo validation with established biomarkers. Now, as I mentioned, the clinical development plan is really interesting because with Parkinson's, we have very well-established biomarkers, some really interesting ones in development. And moreover, by phase 1B, we can already look to measure acute disease reversal with some of these biomarkers. And that means, again, time to clinical proof of mechanism is even less that most other companies will be able to establish safety and tolerability. So here's the team. There's myself and 2 other founders. Each of us have more than 20 years of R&D leadership experience in big pharma as well as in biotech. For example, Daniel was formerly head of translational medicine and neuroscience at Roche. I've previously formed companies in this space, Synexis more than 20 years ago. And yeah, we're looking for 2 or 3 million effectively pre-seed funding to finalize the selection, optimization, and full validation of our preclinical drug candidate for Parkinson's disease. And based on the interest to date we get from investors, we'll know we'll have a queue of investors at that point. And also pharma companies, but we need this 2 to 3 million to get to that point. And that's it. Thank you. Any questions?